Did you know that light can have a huge impact on our body and health? Not only does it influence when and how well we sleep, but it can also affect our mood, alertness, and even our behavior. Welcome to Light O'Clock. In this podcast, we unpack the science behind body clocks and how light influences our daily biological rhythms. In each episode, we chat to experts in the field and have them break down the scientific topics so you can be enlightened. With this episode, we're reaching the end of our first season of Light O'Clock, and so for this reason, we're wrapping up with a special episode. This special episode is part of a feature which we call Shedding Light On, because, of course, we would not be happy without a war pun. In the Shedding Light On series, we'll be taking a deep dive into a specific study or work of a guest on a topic which is relevant to what we covered in the season. So today, we're shedding light on how pupil size changes across the lifespan, and we're joined by Rafael Lazar. Rafael is a PhD student at the Center for Chronobiology in Basel, Switzerland. He's interested in how light can be used or not be used to enhance sleep, performance, and well-being, and what we can do to live a healthy, sustainable lifestyle in a postmodern world. Rafael, thank you so much for joining us on the podcast, and in fact, for closing up season 1 of the podcast. This is our last episode of season 1 of Light O'Clock. Today, we're here to talk about a brainchild of yours, which is a study that you did on pupil size and how pupil size changes across different people and in the field in particular, so in real-world scenarios. Now, we already talked on the podcast in episode 2 about how the pupil is sort of this portal through which light enters the eye and enables us to do all kinds of things. So enables us to see things, but also enables us to synchronize, for example, our inner clock, so our inner biological rhythms with the external light-dark cycle. Now, I think it would be good to sort of start by describing other things that the pupil can do. And one thing that the pupil can do, which listeners I think are familiar with, is that pupil size can change a lot according to different light conditions. Do you maybe want to explain how that works?
Yes. Thanks for having me, first of all. And I'll gladly explain a little bit. I think people are familiar with that, as you said, with the idea of that when you go into bright light, the pupil can be much smaller than when you're in darkness, then the pupil could expand. And that's basically it, the pupillary light reflex. The pupillary light reflex is also thereby controlling how much light enters our eye, as you said. So it's quite obvious that under dim light conditions, your eye wants to get in as much light as possible, so you're able to see. While under very bright light conditions, your eye protects itself against too much light. And also you don't want to be struck by too much light on your retina, so you wouldn't be able to see as much as if it's the exact right amount. So that's what the pupil mainly does for our visual system.
There are also many other things that change the size of the pupil, right? So one classical thing that people can realize is that drugs also affect, for example, how pupil size sort of varies.
For sure. I think that's also a fact that many people know maybe. And that's why police also screens drugs like that, right? There's like police. I have been screened before, actually. There were people would shine a light into your eyes and look how fast your pupil reacts, for instance. And if it stays big for a long time, that would be a sign that you maybe would have taken certain drugs. There's also many other smaller effects on pupil size. For instance, there's the accommodation effect. So when you look at something that is closer, for instance, then your pupil size would change. It delays from near to far when you look from near to far, and it would constrict from far to near.
To sort of enable us to see better, right? So like to focus on different parts of a scene, for example.
Exactly, yeah. So that's also mostly for vision. Then we also have other processes that are actually used in a lot of in a lot of projects and experiments where we would hold the light constant, so we don't have a pupillary light reflex that we just discussed, but we would see other effects of pupil size. So there's, for instance, effects of recognition. So when you recognize someone, your pupil will dilate a little bit, or for certain mental effort. So when you are really engaged into a task, then you would have a dilation of the pupil. And also for emotional arousal. So the sympathetic innervation in the eye would expand your pupil shortly. And that is used in science quite a lot.
So that's kind of when you're feeling a bit anxious, you would have a bigger pupil?
Yes, at least for a little while. It wouldn't be a constantly bigger pupil necessarily, but there is a fact of, for instance, you see something that makes you anxious, then you would have a little like expansion of your pupil, basically.
And let's get a little bit into more detail in the work that you did. So you set out with your experiment to test how the pupil changes, so how pupil size changes in the real world scenarios and also in different people of different age. Now, maybe before we go into the real world scenario, why is it so interesting to look at differences in pupil size between people? Why is that important to look at?
Yeah, so we talked about the pupillary light reflex at the beginning, right? So our pupil, by this reflex, basically determines how much light our retina can receive. And our retina contains photoreceptors that, for instance, enables our vision. And in that case, by letting in less light, we would, first of all, influence our vision, which is very important. But as you mentioned briefly, it also determines how much light gets in for other processes. So I think in a prior episode, you talked about these non-visual effects of light, right? So for instance, melatonin suppression in the evening when we have artificial light is determined by the amount of light that gets into our eye. And because pupil size can regulate that to quite a large extent. So it's, I think, around 16-fold changes that our pupils can create. So when you have a 2 millimeters small pupil from the diameter, you let in much less light than if you have it fully dilated at roughly 8 millimeters. And by this route, many processes can be influenced. And in our case, I think we wanted to see under the conditions that we live and move in, it's important to know how the pupil behaves without looking at a specific process. So we're not looking at like, how does this emotional picture influence our pupil, but we want to know the baseline. Like when we just move around and live our life, how's the pupil behaving? And if it behaves differently across people, then there might be certain things that we need to take care of. For instance, maybe I can also briefly mention this here already. There's an effect of aging. Maybe people are familiar with this as well. So older people don't have pupils that expand as much anymore. And it's not known if this is really as much the case in real world conditions, but what we suspect is if there is an effect, I mean, people who are old and have pupils that stay smaller, they actually notice this because their pupils don't adapt as fast either. So if they go into a dark room, maybe you know this as well. Like I remember my grandma always turning the light a little brighter than I would have wanted it in the evening because she said it was too dark. So this kind of process where you realize, oh, there's a difference that actually makes a difference in the real world as well, because you need more light to be able to see and not fall maybe as an old person. So that's why it's important to also see, hey, what kind of differences are between people, how light sensitive are different people, and where do we maybe need to be careful that we, for instance, for melatonin suppression, since you talked about this already, the idea would be to dim down light very much, but maybe this is not the case for everyone. Maybe we need to be careful because people are different.
Thanks for summarizing the importance of why we have to look at different pupil sizes between or among different people. Now, going back to sort of your study, you specifically looked at this in the real world, as you said. If I understand correctly, this is sort of the novelty of your approach. So before, people had looked at this mostly in labs, right?
Yeah, that's correct. There are a couple of studies who looked at outdoor pupil size, but I think it's important that we note that just a picture of the eye under outdoor conditions is not necessarily reflecting our real world habitual living style anymore, because we do spend a lot of time indoors. We do spend some time outdoors. We do move around. We use screens. We do all these kind of things. And our goal was to look at the things that we know from the lab where it actually matters most, and that is under real world conditions. To an extent, of course, we cannot simulate all the different real world conditions, but we wanted to have at least an amount of things and not just a static setup. So many lab studies, for instance, in the past, where many important findings were found, people would have measurements where people would have a chin rest, the participants, and then everything would be very standardized, so you could really find out what happens without having all the noise of the real world, basically.
Yeah.
But now that we know from lab studies that certain effects are present, we wanted to see, does it still matter if we do these effects matter, and do they still happen, or are they detectable when we go outdoors, when we just sit in front of a screen, when we go from dark to brighter environments, all these kind of things.
Yeah. To sort of lead into what you wanted to find out, we now talked about these lab studies that sort of establish that the pupil reacts differently to different types of light.
Mm-hmm.
Something you wanted to look at is whether pupil size also in the real world is determined by something that we call melanopic EDI.
Yes.
I think it would be good if we try to unpack this for listeners, because it's quite a mouthful if you say the whole name. And I think it is actually really useful also for future episode of the podcast.
Sure. So melanopsin is this photopigment that I think you discussed in the last episode as well, right? So melanopsin was found in a certain type of cell that is in the eye, that was known that it is a cell that is important for pupil size because it summarizes also the signals from other photoreceptors. So from our vision photoreceptors like rods and cones, people might know that from biology back in the days in high school. Those affect pupil size, but people found out that pupil size is also affected by these cells that not only sum up the signal, but also have their own photopigment, so their photoreceptor themselves, basically. And this photoreceptor, or these cells, are called IPRGCs in short. And IPRGCs stands for intrinsically photosensitive retinal ganglion cells. That's also a mouthful. To break it down, those are the cells that contain melanopsin that we are interested in. When we talk about the cones again, so these color vision photoreceptors, those are also influencing pupil size, but the very brief and quick effects. But the maintenance of the pupil size under constant light conditions, which are natural conditions. So, I mean, when you walk outside, there's either light or it's dark, but it usually doesn't change very quickly. This level of pupil size is maintained mostly by melanopsin and the IPRGCs that contain melanopsin. So that's a very long-winded answer, but that's why we're interested in those effects, first of all, right?
Yeah. Thanks for clarifying that. I guess if I were to rephrase the question, so now we know, so as you said, we know also from lab studies, right, that pupil size is determined by IPRGCs, or let's say IPRGCs influence pupil size. Now, how do you quantify that? How do you quantify how IPRGCs are responding to light, and then how that in turn affects pupil size?
Right. Yeah, that's a very good question. I think what you can do with any light, you measure it. You can quantify it somehow, right? You can, you have different wavelengths, and you can just measure that. But a lot of times, what we already do is when we measure light, we adapt it to us humans. So maybe people have already heard about the unit lux, and photopic lux it's called, or illuminance, is basically quantifying the light against the sensitivity of two types of the cones that the eye contains. By that, we already basically anticipate how much the light would influence our cones. And it's a very good unit, this photopic lux, because it already quantifies basically in general how much we would perceive it as bright, because those cones, L&M cones, those sensitivities summarized basically give like a function that explains how bright we would perceive the light as. So it makes really sense to measure that and to say, well, the light is basically 100 lux bright. That means we would perceive it as very bright or not so bright. 100 lux would be like medium bright, I guess, and 1,000 lux would be really bright for us. But that wouldn't account for the non-visual effects that happen with that light, because that light might not contain a lot of blue light. And then if we say 1,000 lux, but it's very orangey and doesn't contain a lot of blue light, it would not influence maybe our sleep and our pupil size as much as we would think. So it's really important to quantify light with the photoreceptor sensitivities. That is the most important in that case. And that's where melanopsin comes in again. So when quantifying the light with the sensitivities of melanopsin, we basically have a unit like melanopic EDI where the sensitivity is already in there. And then the EDI stands for equivalent daylight illuminance. And equivalent daylight illuminance just relates that to daylight. So maybe again, an example maybe makes it simpler. So when we talk about 1,000 lux melanopic EDI, we talk about the amount of light that would be contained in daylight that activates melanopsin. So 1,000 lux daylight would activate melanopsin as much as that specific light that we talk about. So if I say this light contains 1,000 lux melanopic EDI, someone knows, okay, so this light is activating my non-visual effects of light as much as 1,000 lux daylight would. And it sounds quite complicated, but it's also kind of getting used to the unit, I guess, but everyone can relate to daylight. So I think it makes sense to also relate it to that as well.
Yeah, thanks for that. Absolutely. And so I guess to understand if I really got it right, we basically want to, with melanopic EDI, what we're trying to do is characterize light as not the spectral composition or how it is, but rather how that light affects us in terms of the non-visual effects, right?
Exactly. Yeah. Very nicely phrased.
Great. And so from the lab, we know already that melanopic EDI predicts the change in pupil size. Is that correct?
Yes, specifically under steady state conditions. So where our eye would be light adapted and this light would continue for a while. So continuous light. Whereas when we had flickering, very fast flickering light, that would not maybe be the best predictor. But that's not natural conditions either, right? If we have flickering light.
That's basically why then you wanted to look at this specific, let's call it measure, melanopic EDI in the real world, right? So you said, okay, this is known from lab studies that this determines the variations in pupil size. What about in the real world when we're surrounded by light? And also the real world is really where the non-visual effects are very important, right? Because, melatonin suppression and all these things, these are things that happen constantly in our biological rhythms daily.
Exactly. And I think one other thing is that there was a gap in the research that we found in the sense of people know from experimental studies that this has an effect, but still there's a lot of models that, for instance, great models, by the way, where you can predict how big your pupil size would be if you have a certain age under a certain condition and a certain brightness of the light. But this exactly uses the amounts of light or the units that we just discussed are better predictors for brightness perception and vision. So there are great models, but they're not basically updated to this new unit. And obviously, we need to establish first that this unit is actually really controlling pupil size in the real world. Otherwise, it wouldn't make sense to adapt any models, right? So I think this was also a reason why you thought, I think we need to measure this. And my supervisor, Professor Manuel Spitjan, he back a couple of years back, he motivated me to go and do that research and answer that research question.
So now that you mentioned that you went out to measure this and to really answer this research question on how pupil size varies in the real world, how would you go about to do that? So did you just gather a group of people, and how did you actually measure their pupil light when they were doing their daily activities?
So that's a good question. I think that's basically why we didn't have a lot of studies like this in the past is the equipment has to be special. So first of all, you have to obviously measure light. And because we wanted to later on in one of the questions that we had, we wanted to know if melanopic EDI, this new unit, compares or like better predicts pupil size than so I call it old unit now, like the brightness perception unit, photopic lux. So in order to do that, you would have good light measurement equipment in order to actually see the differences. So we used a spectrophotometer, spectroradiometer, that is placed on the forehead, basically. So we had to think about a solution of where to place it. And obviously would like to place it directly in the eye, but that's not possible. So we place it on the forehead, try to adapt it in angles so it fits roughly the eye. And then also we needed to measure the eye at the same time. So we had an eye tracker that would take pictures of the eye. And I'm still a little sad about that we don't have like really good time resolution and no video-based recordings, but we did manage to have a Raspberry Pi computer that is just like a little small computer that you could program, and that would take measurements at the same time of the light and the pupil. And this would enable us to make people move around freely, which is the new part of the study, basically, because other studies would just, even outdoor light studies, would just place someone in outdoor light and then take pictures of the pupil. But we wanted to be able to move around. So that required special equipment.
Right. So basically they were going around with this spectrophotometer on their head, if I understand correctly, like a headband sort of.
Yes, exactly.
Setup.
Mm-hmm.
And this was measuring the light that they were exposed to.
Mm-hmm.
And then the glasses, the eye tracker was measuring the pupil and how the pupil size was changing.
Exactly. Yeah. I should have mentioned this is an eye tracker that looks like a pair of glasses without the glasses. And then it has like a little camera on the side of the eye. So it doesn't disturb your vision, but you would see the eye. You could record the eye, basically.
Right. And how long were people actually wearing these glasses or this setup?
Yeah, I would have loved to do longer studies, but it wasn't bearable. So we ended up with a 1 hour protocol. 20 minutes of this was roughly, we also had a dark adaptation. So we wanted to know where's the baseline pupil. If you dark adapt them for 10 minutes, you're probably able to see the maximum pupil size because it takes a while. The last few half a millimeter, I don't know, like the last few bits of the dilation process takes a lot of minutes. So 10 minutes is roughly, I think, a good amount. And then we had a lab study part where we would have certain artificial lights where we know that blue light, maybe it stimulates it more than red light. So we would have that as a confirmation, basically, just as we know that we measure the right thing. And then we would go and have our real world scenario where the other 40 minutes of that study, people would do indoor and outdoor tasks. And we would have like a script, what we do with them at what time. And then we would move out eventually and also be an outdoor light.
So they were just also walking in the street with this setup and just going about their daily activities, basically.
So they would start by, we would actually sit then in front of a screen because we had some questionnaires that we wanted to let them fill out anyways. For instance, a rating, how tired they were, and all these kind of things. And we thought we can incorporate this into the real world design because everything is allowed. It's all real world, right? So, so they would sit in front of a screen, fill out these questionnaires. Then they had a task where they would actually read something to us. because we wanted to have different accommodation status, and also just a normal task. You would read something that's a normal task, right? If we were walking around indoors with them, we would ride the elevator with them. They would also look out the window for like a minute. So we would also time this to be a minute roughly, because our measurements were done every 10 seconds. I should have mentioned this earlier, I guess. But so the spectral measurements of the light as well as the eye measurements were taken every 10 seconds, which is quite a low time resolution for our case. But because we're looking at continuous light conditions, this should be fine. And also another reason why we did take roughly a minute at least for every light condition, because we wanted to measure enough samples for each light condition. So they would look out the window for a minute, we would record this, and they would read something to us, which would take them more than a minute, and so on and so on. And yeah, there are all these kind of things that we try to emulate the life that people would live, at least to a certain extent. And then from the indoors, we would move out for the last 15, I think it was 15, I have to look it up, but 15 minutes, where we also had certain tasks where they would read a sign on the street, or they would look into a little lake that we have there, or we had overlapping tasks. So we wanted to keep the task constant, but move through different environments. So they would again read something to us, the exact same length of the text, but not the same text. And then that would be under outdoor light conditions. So while we had this very open, everything goes real world design, we still wanted to keep certain things constant and look at the same tasks under different light conditions.
This sounds like such a fun protocol, honestly. You're just walking around with these people wearing this really cool setup and them just doing all these different activities.
It was quite fun, yeah.
Yeah, I can imagine. And so coming to your results, what did you find?
So we have three hypotheses, three basically main research questions that we had in mind. And that was, first of all, looking at how light determines the pupil size. So is there like a linear effect or roughly linear effect or like a log linear effect? So would the light have an influence on pupil size? And if we have dimmer light, would they have bigger pupil sizes? So the basic pupillary light reflex. If that didn't happen, then the whole study would be really weird, because this usually should happen. And it did. And then we also had the age hypothesis, where we wanted to look at, well, is there an age effect? And under which conditions is it most pronounced? Is it in the dark condition in the lab? Is it when we have dim conditions, when we walk around here indoors? Is it in the very bright light conditions? And there was already research showing that the effects of age are strongest, obviously, when the pupil is dilated most, so in darkness. But we wanted to look, how does it look in real world, right? And then the final hypothesis, where I was really not confident that we'd find anything, was if melanopic EDI, this new measurement of a new quantification of light, predicts pupil size better than this older quantification of light, photopic lux. And why I was not confident was because the light that we receive outdoors and indoors, it's still, the two units correlate quite a bit. So they're still very similar. So if you have bright outdoor light, both quantifications would be very high. In the lab, you can manipulate it. So you can have blue light, which is very strong in melanopic EDI, but very low in photopic lux. And then red light, which is exactly the opposite. But when you have real world light, you have basically just white light. And you have obviously some reflections of objects. So when you have a red object, that would also give you more red spectra into your eye, more red light. But at the same time, the correlation, and we looked at it, is super high. It's like 0.999, I think. So it's like almost 1. And that means that the lights fluctuate in a very similar manner. So that's why I was a little, not very sure if we'd be able to find, under those not controlled conditions, that we can still predict pupil size better with melanopic EDI.
Yeah, very cool. And going back to the age hypothesis that you had, so this idea that, okay, do we see age effects also in the field? What did you find there?
So as expected in the dark conditions, we would see large age effects actually. What actually surprised me a little bit is the inter-individual variation, meaning that people, even in the same age group, are so different. But when looking at the literature again, it shows in many studies, and that's actually also discussed in many studies. So, for instance, we would have, and I think it has to do, this is just a guess of mine, but we had not so many people over 65 as we wanted. We did have a lot in the middle, and you can see the age effect quite clearly. For instance, now when we talk about very dark environments, so in the dark adaptation, you would see a clear reduction of the maximal pupil size. But then in the end, where we only had like 3 people over 80, you would see that because of the large individual differences, 2 of the old people actually had still large pupil sizes and only 1 would fit that. But if you statistically look at it, there's quite clear a good linear age effect.
Meaning that older people would have, as you said at the beginning of the episode, would dilate the pupil less,
yes,
right?
Yes, exactly. And I can actually say, I think I have the numbers in my head. So it's like per decade, per 10 years, the maximal pupil size would shrink by 0.4 millimeters. So it's a half a millimeter of pupil diameter per 10 years, roughly.
Which sounds maybe not like a lot, but if you think about how big the pupil
is,
it
is quite
right?
a lot. And also if you go from age 20, if you compare age 20 to 80, you have 6 units basically of decades. So almost 3 millimeters smaller pupil size in a person that is 80 years old. And that would make quite a difference. So you could also calculate, if you have the light measurement and the pupil size, how much light actually can enter it roughly. And that would make quite a big difference.
And in terms of how to take this result about age into applications for the real world, we often now talk about human-centric design, or sort of thinking about light indoors as well as something we also have to consider for our circadian rhythms. How do we put this finding of age into a framework? Would that mean that older people need different kind of lighting compared to younger individuals?
Yeah, I think you mentioned a really important point there. I think we need to think about what does it mean. For the age effect, we still found the age effect under bright light. Maybe I should mention this, but it gets smaller. So with every step of increasing the light, the age effect is just less pronounced, but you still see it even under bright outdoor light. And I think what this means, especially under the dim light conditions, which is not surprising because we know this from lab studies, but since we find this under real world condition as well, is the pupil size is really something that declines with age. We need to be careful not to over design our human-centric light, I think. Human-centric lighting is a wonderful idea of like adapting our light to different times of day and changing it accordingly. So our bright light, we get enough bright light during the day and not too much light in the evening. But the problem is older people might have problems to see in dimmer light. And also the pupil not only gets smaller, it also gets slower, which we didn't look at here, but still it's really important that we are careful not to over design it so that people may even fall or something. Like an 80-year-old person, you need to be careful not to make them fall because it's too dark. And I think just showing this in the real world again is a reminder that we need to eventually individualize this light things. And sometimes people also know what's good for them. I know bright light in the evening is not great, but if my grandma wants to have brighter light than I do in the evening, maybe it somehow makes even sense, right? And
yeah, absolutely.
I think that's the main takeaway for the age finding.
Yeah. Just to wrap up, if you were to look further after this study, what would you look at? What would be interesting questions that you are curious to ask?
Wow, there's a couple of things, but I think the main thing that you could expand this study specifically on, I just mentioned that the pupil gets also slower in age. you would have to do this with better time resolution. So I mentioned that we took still pictures of the eye every 10 seconds, and you wouldn't be able to see smaller changes of pupil size. In this way, you would also maybe not detect the cone mechanisms, or like the cone, the photoreceptor that drives the quick changes of pupil size. Maybe you can also test that again to some extent. So I think if you really want to dig in the how more, you would need better time resolution. And the great news is the technology advances so quickly that right now we already have affordable eye trackers that work with Bluetooth, that don't have any cables anymore. So you would just need to somehow make those devices like the spectral radiometer, as well as the eye tracking devices, to measure at the same time, or like you have the same time points that you measure. And then you could basically just redo the study and look at other things. And also, I think pupil size is just a thing that is rising quickly. There's so many possible applications where pupil size comes in handy. It's used in marketing for like gaze direction, where people look at most. It's useful for quantifying how much light gets into your eye. It's for detecting arousal or emotions. So I think pupil size in general already is sparking a lot of interest. But now with the devices that we have that you can just wear like normal glasses, this is going to explode, in my opinion. And I'm actually happy about it.
Yeah. Well, thank you so much for unpacking what you did, this very important work of studying pupil size in the real world. And very much looking forward to seeing this work published at some point soon.
Thanks. Yeah, I'm working on it. So stay up to date. I'll send it to you if it's out there.
Yeah, absolutely. Yeah, and depending on when the podcast episode is coming out, we'll link it in the show notes.
Perfect. Thanks for having me.
Yeah, thank you so much for coming and for the nice chat. Many thanks to Rafael for joining us and sharing his work on pupil size variation in the real world and across the lifespan. Rafael's work has now been published as a preprint, so you can go and take a look at all the beautiful graphs and pictures of the pupil that he has there. You will find the preprint following the link in our show notes. To our listeners, thank you for tuning in into this episode of Light O'Clock and actually for sticking with us throughout our first season. We've now come to the end of it and we'll take a short break. But fear not, we'll be returning with the second season, where we'll be delving a little bit more into circadian rhythms. But I don't want to give too much away, so if you haven't already, just make sure to subscribe to the podcast and you'll know as soon as season 2 is out. Until then, well, you know it. Don't forget to lighten up your clock.